Hello, everyone. It is a pleasure to introduce Dr. Valentina Fossati in here today. Personally, I worked in her lab in 2016, right before coming to Georgetown. She's such an inspiration to me, and I am so excited to hear about her work here again with all of you today. Dr. Fossati is a senior investigator at the New York Stem Cell Foundation, where she focuses on advancing preclinical studies of neurodegenerative and neuroinflammatory disorders, utilizing induced pluripotent stem cells derived from the patients. Dr. Fossati received her undergraduate degree in pharmaceutical biotechnology and her PhD in stem cell biology from University of Bologna. She did her postdoctoral training in the laboratory of Dr. Hans William Snook at the Black Female Stem Cell Institute at Mount Sinai. In 2011, she was recruited
I think you're frozen.
We lost Jingwen.
Jingwen, you're frozen.
She'll come back.
Probably knows that.
Sometimes you don't know.
Wow.
This is when the rest of us like peered quickly at the screen though to see if like anyone else was blinking. So was it us who was frozen or was it like the speaker? Because you just never know.
But she chatted. I will come back in 1 minute. So I don't know, maybe it was before.
Well, this is a good chance for you to tell us what she was like.
She wrote a good reference.
No, she's very sweet, as you said, and she was very excited and very passionate about our work, really keen to learn everything we could about iPSCs. And it's not an easy summer internship because it's not much that you can do in a summer. And she came when we had the smaller lab, and it was a completely different lab also for me. But at that time, so much change in the past years. So, but I'm happy that she continued with the PhD, and she's really continuing with this science work. It's the most rewarding thing when she wrote me. Actually, she invited me here. It's really nice when you mentor people even for a short time, and they remember you and they call back. I think is really the best reward that you can get.
Oh, I can see her face.
It looks like we're back.
That's so sweet. Should we start or can I finish the last one paragraph?
We can start, whatever.
Great. Dr. Fossati, the last paragraph. She established, that is really impressive. She established protocols to generate oligodendrocytes, astrocytes, microglia, and neuronal cell types. And now she is developing organelles and co-culture systems to identify and target the key pathogenic mechanisms leading to neurodegeneration and demyelination in progressive MS, Alzheimer's, and other neuronal disorders in the CNS. Let's welcome Dr. Fossati to start her talk on iPSC-based models of glial cells for studying CNS diseases, present and future perspectives.
Thank you so much, Jingwen. That was perfect and very, very kind of you. And I will share my screen and see if you guys see everything. Do you see my first slide?
Yes.
Perfect. So we can start. And I saw in the emails that we try to keep it, really as for more lay audience and not really people expert on iPSCs. So it's always hard to understand how much people know. So please feel free to stop me if something is not clear or if it's too much clear, we can fasten up. So really, let's make it this a very interactive discussion, pleasant for everyone. So as Jingwen said, I work at the New York Stem Cell Foundation Research Institute, and I just have one slide because not many people are familiar with us, especially outside New York. This is a foundation that started really as a foundation, meaning getting philanthropic money and providing this money for postdoctoral fellows in 2005, when the Bush administration banned federal funding for embryonic stem cell research. So the goal was really to promoting embryonic stem cell research and making sure that the young generation of students and postdoc would not quit the field. But then very shortly after, we actually became our own institution. And so we started with a small lab in New York City up at Columbia. And then we expanded, and we are now, this is New York sound, I'm sorry. And we are now over 100 scientists in Midtown, and we have our own institute. So as I said at the beginning, in 2005, when myself started, we were working on, they were working on embryonic stem cells, and I was in my postdoctoral lab. And so, as you know, we could use embryonic stem cells isolated from the inner cell mass of discarded embryos. And these cells are pluripotent, and that can be used to make all the different cell types of the body. But there are ethical concerns because the use of embryos. And so that's why there were several bills along the years, even past the Bush administration, that are really, really blocking federal funding. And so the research on embryonic stem cells is still quite limited in US. But meantime, in 2006, Dr. Shinya Yamanaka made a groundbreaking discovery that really turned the field of stem cell biology. When he showed that you could take, first he did with skin cells, and in 2006 he did with mouse, and then 1 year later with human cells, you could turn those adult cells into stem cells that he called induced pluripotent. And they can basically be generated in a dish, and they are equivalent to the embryonic stem cells. This was obviously very important because it's removing all the ethical concern, but also for the students here today, you need to understand that this was really going completely against the dogma. When I and your mentor studied 10, 20 years ago, we were taught that this never happens. This is science fiction. Once a cell becomes adult, it cannot revert back. So it was really a major discovery, and not surprisingly, Yamanaka won the Nobel Prize only 6 years later. But the other very important part of this discovery, which made those cells so much more appealing than embryonic stem cells, it's not only that there is no ethical concern, but for the first time, you could work with live patients. So you can recruit patients and ask them to donate a skin biopsy, although now the field has moved really much more with blood because it's much easier to donate blood, and patients have to do it anyway most of the time for their exams. So we ask a few drops of blood from the patient, we convert them with the stem cells, and there you have in a dish the possibility of making all the cells type that you want to study, depending on your disease, with all the genetic of that patient, which is something that, of course, you couldn't do with an embryo, which was, of unknown identity. So this has really, completely changed the field. And also at myself, obviously, we have largely moved working with iPSCs, although we are still one of the few labs that is still working on embryonic stem cells. It's been a decade since then, so the work on iPSCs has progressed quite fast, I would say, considering that the time that you will see later for the work. And so what NICEF has been actually doing and leading is expanding this work and making it more large scale. And so we built a fully automated platform that is using robots to do all the steps of the generation of the iPS lines, from the skin digestion or blood, PBMCs isolation, reprogramming, expansion of the colonies, freezing, thawing. And also this platform allows to make hundreds of lines at the same time. And so we have made a few thousand iPS lines now. We work in partnership with big universities or pharma, and they come with us with a cohort of, let's say, 400 patients For example, there was a project with Monsignor on schizophrenia patients, and all these lines are made with the robots, and we can clearly see that it changes very much the quality of the line, because no matter how good you are when you are manually doing something, there is always, something that can change. Maybe one morning, you didn't drink your coffee, or there is always something that changes when something is artisanal. And so also with making the cells, having a completely automated production really decreases the variability line to line. So this is, inside all this context, when I joined ISAF, I decided to work on working primarily on neurodegenerative diseases. And so we have other colleagues. And so one big part of our work in ISAF is to work neurodegenerative diseases. Why? Because, as you know, those are really very, the hardest to treat. They are a major problem of our times, mostly because the population is aging. And so, they are only going up. If we look at the perspective in 10, 20 years, and we have several million people affected, and we don't really have cures. And a lot of these diseases have animal models, but they are models obviously with their limitations. So those are exquisitely human diseases. And so the possibility of creating also human models, we believe it's very important, and will bring a completely new perspective and a completely new tool complementary to the animal models that you have to be able to understand these diseases. And so what I did since I joined ISAF, really exactly 10 years ago, was really creating still the building blocks. And so making sure that we had the protocols for generating the cells of interest. And I always had a particular focus on the glia cells rather than the neurons. And so we have spent really a lot of time in developing and optimizing protocols, also confronting with other independent groups that have been done the same during these years. Only now, reaching at the end and being finally able to do the more interesting and more translational studies where we can really apply all these protocols to specific diseases. And so I will go with you today through this journey and the way that we have made the cells. I just want to make sure, to highlight that again, we always talk about models. And so this is something to be remembered, because like animals are a model that reflects somehow some aspect of the human diseases. In this case here, we are making cells in vitro. So we always have the question, are these cells equivalent to the primary cells in the brain? Obviously, they are not the same. It's a model, but it's a model that has these pros and these cons. It has these weaknesses. So we have to be very honest and very aware of the limitations. And I think, if we are aware of the limitations, we should be able, and we really should, build the proper experiments that allow us to answer the proper questions. So when used correctly, I think that this model can be very powerful, and hopefully we will see in the next years many pathogenic mechanisms discovered through iPS modeling. But again, we have to be very careful and sometimes not abuse of this system. The way I'm specifically working on this in the lab, it's in collaboration with another PI, Scott Noggle, and we are part of a large consortium with mostly people at Mount Sinai, but also another university that is led by Bin Zhang. At Mount Sinai, Bin Zhang is a really bioinformatician and has been driving the first transcriptomic data, getting brains from Alzheimer patients. They started with transcriptomic and then they moved on proteomic, metabolomic. So we're trying to get all possible data from these patients. And what they identified from those screens, from this analysis, was potential key drivers. So some genes that were either upregulated or non-regulated in the Alzheimer brains. Interestingly, all these genes that we found are primarily expressed in microglia and astrocytes. So what we are doing now with the postdocs in my lab, Chandrika is working on making all the edited lines. Once we have the edited lines, again, I think this is the best way that we can use the IPS model, because you have a system where you have your line and then you have a knockout of the gene, or you can use CRISPR AI, so you can activate the genes, you can repress your gene, and then you can look at the role of the gene in the cell type. And so the idea is really to hopefully identify new pathogenic mechanisms which would lead to new therapeutic strategies for those diseases. And so, we are making, once we have these lines, we make it the different cell types alone, we combine them, the cultures. The idea is to obviously have the mismatch where you have a wild type neuron versus CRISPR edited microglia or similar. And then we also have the organoids culture, which we talk a little bit at the end, which became very popular in the last 3 years, I would say. So let's go through, all the different protocols that we have developed. The first one that first really work that I started, even chronologically, it's on oligodendrocytes, because my long-term interest and goal is to really understand the process of remyelination for developing remyelinating strategies for MS. And this was my work with my first postdoc, Panos, which Jim met and had the chance to work with. And now the work has been continued by Maria, which is the current postdoc working on it. So I don't know, some of you are very familiar with MS, maybe some a little less, but very making it very simple. In multiple sclerosis, the hallmark is the demyelination. So there are infiltrating immune cells that attack the myelin, which is the insulation in the neurons. And there's been a huge amount of work focusing on these immune cells, why these cells are infiltrating, how can we stop this infiltration. There are now over, I think we reached over 20. Yeah, sorry, this slide is, there are 8 first-line therapies here. Maybe this is the number, but we have about 20 now drugs available. And all these drugs are really immunomodulatory, so they are targeting in different ways, but all the immune cells. But still, what the challenge is to come up with some complementary drugs that are working on improving the myelin and somehow restoring the myelin that is lost and helping the brain in the patients that become progressive. So the patients that have this major neurodegeneration occurring, so how can we restore their myelin? So we don't have yet these drugs, and many laboratories are now having this as a priority in their studies for MS. So the next goal is really coming up with these regenerative strategies. So for doing that, we are trying to work with the human iPSC-derived oligodendrocytes. And so this protocol we published now has been quite a few years. Many years have passed, but really not much has changed. And I still believe that oligodendrocytes are the most challenging cell type to make from iPSCs of any other cell type. They're very, very frustrating sometimes, but on the other side, exciting. So if you have read papers and also recently of iPSC differentiation, there are 2 major schools and 2 major methodologies. The first one is the one that I've used, that is the most traditional, that started at the beginning, which is we try to feed the cells with the molecules that recreate the microenvironment that the cells have during the embryonic development. So in the case of oligodendrocytes, we know from mouse studies, from mouse embryonic development, that there is this PMN domain in the spinal cord where both oligodendrocytes and motor neuron arise. And we know that there is a specific concentration of retinoic acid and sonic hedgehog in this domain. And so this is what we are trying to, we feed the cells with. And before, you see here in the first step, it's SBLDN. This is the shorter name for the 2 small molecules that are dual SMAD inhibition. They're inhibiting TGF-beta and BMP4 signaling. And this was a very important findings from Lawrence Suter, which is one of the pioneers in all the neuronal differentiations from iPSCs. He saw that when you inhibit these two pathways from pluripotent stem cells, you massively drive the cells towards ectoderm. So this has now been used in every single protocol. If you want to make any type of neuronal cells, neural cells, you have to go, you start with this, what we call, the dual SMAD inhibition, this SPLDN small molecules. So the advantage of this method is that you are basically recreating in vitro what, mimicking really what happens during development, but this can be very long. So in the case of oligodendrocyte, the cells really seems to follow their internal clock that they have in the human brain, and they come up really as the last one. You see our protocol is about 75 days long, so you have these first neural spheres. We play them down and we let the cells grow out of the sphere, so cells migrate out. And first you have all the neurons and astrocytes. The astrocytes create a nice layer, and really only at the end, OPCs and oligodendrocytes appear, which really seems to mimic what happens in development. So we haven't found yet a way to generate a plate of full just oligodendrocytes, which interestingly is something that you can achieve much easier with mouse embryonic stem cells. If you use mouse embryonic stem cells, in a couple of weeks, you can have 80% OPCs and, really nice, and the OPCs are able to expand. With the human cells, it's still much more difficult. In fact, you see here at the end of the differentiation, we have some MVP positive oligodendrocytes, but they are in the middle of all the other cell types. With those cells, we have seen that they are myelinogenic. So in vitro, with these neurons present there, they are not really able to wrap. But if you isolate them, we sort them and we inject them into Shiverer mice, so mice that don't have myelin, and they are also immunocompromised, so they can accept human cells. You will see myelination forming in the brain. The other way that you may have read papers about making mature cells from iPSCs is through transcription factor driven protocols. So in this way, we are overexpressing really important critical transcription factors to the cells, and then you're forcing basically the cell to become whatever you want. And recently, again, Lawrence Suter published a protocol for making astrocytes, and neurons are made this way. There are also a few protocols for oligodendrocytes. The advantage of this system is much shorter. Usually it's half of the time. So you have the cells within 30 days, maximum 40 days. And you can achieve a higher purity of the cells. But when you look at if you look back at these papers, you see that the morphology is not that great. And I still have not really understood, how much is that important, and, how different are the cells. So we don't have yet many comparisons. I think that a lot will come out. We are also trying to do that. But it's also a different system. Again, you have to remember what is your original question, because when you overexpress transcription factor, you are forcing the cell to go to that direction. And so the limit could be that maybe, if you're studying a disease, you may miss a phenotype that instead would show off when you have this protocol that is more like natural and goes on for longer time. So depending on what's your need, you can use one of the other strategy. And so we have tested our oligodendrocytes, and Maria now spent quite a long time, and now thanks to the lockdown that she was working from home, really working on creating a pipeline for quantifying the oligodendrocytes in an automated way, which is not that trivial, because you see are really highly ramified cells, very complex. So you have to be able to distinguish the different cells, count when they are attached. So it's really not that simple, but we made it able, and we have this Opera Finis, which is an automated confocal microscope that is really suitable for using as this screening for screening purpose. And so we have tested some myelinating compounds that have been discovered recently. And for example, we have a long-standing collaboration with Paul Taser that did one of the first screens for myelinating compounds and identified a few years ago miconazole that was originally in the paper, which is interesting because it's an antifungal drug and nobody really thought that would also have myelinating properties. And then from there, Paul has started his own company and is working on making it as a drug that is more, really deliverable in the brain and really can be taken by your patients, because miconazole as it is could not be used for MS patients. And so he found other compounds that are all working on the cholesterol biosynthesis pathway, and tazein is one of them. And so this is the one that we have been using. And you can see here the comparison, even just visually, when you treat your cells with tazein 100 nanomolar, you have an increase in the production of the number of oligodendrocytes, MBP positive oligodendrocytes. So Maria is now working on developing a drug screen for myelinating drugs, or for drugs that support oligodendrocytes survival. And in this case, as you see here, the caveat of this protocol that we don't have pure cells, and so we are working instead of the overexpression of transcription factors, trying to actually make a pure population of OPCs and be able to perform the screen on really, much, almost pure population of OPCs. So switching gear, do we have no questions, Jingwen? So good to go?
Yes, there's no question.
Can I ask a quick question?
Sure.
So it's very cool. Your previous slide, you showed that you're screening in primary progressive and secondary progressive patients, I think.
Yeah.
So how many patients iPS cells do you need to use for you to be certain that your drug is actually promoting remyelination and may be effective for the treatment of that particular phase of the disease?
Yeah, this is a proof of principle experiment that we started. We have these lines. Our cohort right now is of 30 patients, so it's very limited. We also made a cohort of patients for Bibi Bielekova and IH, and we are working on 2 more cohorts with Peter Calabresi, Johns Hopkins. So the idea is to increase the work. So obviously, what we do first, for the drug screening, we're going to only use 1 line, which is a healthy control line, and that's the idea of testing new compounds. But then, yeah, in the long term, the vision would be, hopefully being able to study more lines. So yeah, so theoretically, you should have, a few hundred lines to be able to do what we call clinical trial in a dish. So as you can say, being able to predict if different populations respond. So which, we will get there in a while, but the first step, we're gonna first have to do this proof of principle experiments that showed. So in this small cohort that we did, I can tell you, yeah, that it's interesting. We didn't find major differences between different group of MS. There seems to be a line-to-line variability, meaning that even between the controls, there are very different responses, and between, so it seems to be more, independent line-to-line variability rather than different groups responding in different ways. But yeah, I mean, and also it's, as hopefully we identified in parallel, other people identified some potential markers, some potential SNPs that may interfere with the drugs. Then that could be that you would require a less, and probably a lesser number of cells, because then you could identify all the cells that have the SNP versus the cells that don't have that SNP, and then how they respond differently. But yeah, theoretically, we hope in the future to be able to move to a huge number. Yeah, when you talk to statisticians, to having a good power, you really need a lot of lines. So it's not there yet, but we go step by step. And as I said, it takes time. 10 years just to get to this stage. So Okay, so the work that we've been doing on astrocyte was led by a very, very young and brilliant student. Lily was 20 years old when she came to my lab. She worked very hard for 2 years, and now she's an MD PhD student at WashU. And she worked on astrocytes. As you know, astrocytes are, the most abundant cells in the brain, are very important to provide several different types of support to the neurons. But lately, there have been, several studies also pointing out their potential pathological role in several neurodegenerative diseases, almost I would say in virtual any CNS disease. So there is this delicate balance that we need to better understand in order to be able to target, what are the negative effects, but being able to keep the positive effects. I said before that, it's very frustrating that we cannot make oligodendrocytes, but on the other side, we try to put it in a positive way. We were always seeing those beautiful astrocytes in the plates, and I said, can we make them, isolate them and being able to study them, because this is so expensive and so long, at least we get 2 cells out of it. And so long story short, we try to purify those astrocytes. And the common marker used for the human cells and also for immunopanning from primary rodent cells is HEPACAM, but that doesn't work well in our cells because our cells really don't express it very highly. And so we did a screen of other old surface molecules available from one specific company to see if we could identify one, and we found a CD49F as a marker that was really able to purify from our cultures all the GFAP positive astrocytes in the positive fraction, leaving in the negative fraction primarily OPCs, progenitors, and neurons. And we have tried to characterize the cells to the best that we could with the tools available. And so we did bulk RNA-seq and single cell RNA-seq. And this is an example of the single cell RNA-seq where we compare unsorted and positive and negative fraction. And you see the blue dots are enriched in the positive fraction on the population that is GFAP positive and aquaporin positive. So really the population of what we call, mature astrocytes. And we seem to have 4 different types of astrocytes in our culture, which we are still, trying to better characterize. And then at the same time, there was also a paper from Sergio Pasca, which is also one of our myself investigators funded. And Sergio and Steve Sloan, they look at the organoids culture, and they saw how in the organoids, it really takes a long time for astrocytes to become mature, over 100 days and even more. And they define this, what they call the early pseudotimes, middle pseudotime, and late pseudotime to characterize the different stages of maturation. And it was interesting to compare our cells with their cells. And we see that in our protocol, which is, is different because it's a monolayer and it's patterned toward the spinal cord. So it's very different, but we seem to achieve the similar level of maturation in a shorter time because our cells are ready between 60 to 70 days. So we try to see what we can do with these cells and how they can be helpful in our disease modeling. And we did co-culture with iPS-derived neurons to find, as we were expecting, is known that they should support the neurons, and we actually see that they are greatly supporting neuronal maturation. So if we have iPS-derived neurons alone by themselves, they're really almost not neurons in the sense they are barely fired. When they are really 30 days after the differentiations, they are very, very mature neurons. But if you put them, keep them 2 weeks on top of astrocytes, you can see clearly how the firing pattern changes and the spontaneous excitatory postsynaptic currents completely change and improve. And then the reason why we got interested in this pathological events on astrocytes was
Can I ask a question about the previous slide?
Yeah.
is that, do you need the astrocytes or can you use conditioned medium?
No, you really need the astrocytes. It's really the co-culture that helps them. We haven't done much experiments with a conditioned medium, but the few experiments that we did clearly suggest that the contact helps.
Thank you.
So the way that we're interested in the pathogenesis of astrocytes was really following up the studies from Ben Barres and Shane Lidlow. And it was kind of a coincidence because Shane moved to New York City, just in the time that we were studying astrocytes. And so it was very easy to set up a collaboration. Basically, I think most of you are familiar with this Nature paper from a few years ago, where he showed that microglia stimulated with LPS releases some various cytokines, inflammatory cytokines, but specifically TNF-alpha-1-alpha-C1q are activating the astrocytes to become reactive. And these reactive astrocytes are secreting something that is directly toxic and killing the neurons. And this, Shane, has been going on, and hopefully it's close to understanding what is this factor, but it's still really unknown on what exactly is this factor that is killing the neurons. And his study was a very elegant study using rodent cells and in vivo and in vitro model, but not human cells. And so I was really thrilled to be able to adapt it and see whether that would work also in the human system. So we stimulated the cells with this cocktail of cytokines. We also saw that we are able to have from microglia secrete all of them. But it obviously is much more complicated because it's much more expensive. You have to make the microglia synchronize, and so for making it much easier, we bought the recombinant proteins. And so even 24 hours after, I think you can appreciate there is a drastic change in morphology, and the cells look much more, like puffier, and now with much more arborization. And they express C3. Which Shane has shown in the paper, identified as a potential marker. And then following what they studied with the rodent cells, we also see if that was true in an in vitro model of human cells. And so they look at the phagocytic capacity so that reactive astrocytes have are less able to phagocytose. We use this system of pH rhodosynaptosomes. So those are synaptosomes that have been isolated from rodents. They are conjugated with pH rhodo. So this is a system that is not fluorescent unless it's in an acidic pH. So if it's just sticking on top of the cells, you will not see it. But when it gets engulfed and is inside the lysosome, it turns into a red fluorescence. And those are the red dots that you see here. So we can quantify them using the IncuCyte. So we basically have this microscope inside the incubator, and we can keep the cells there and take a time-lapse videos. And you see here we use 3 different independent lines of astrocytes, and see that there is a significant difference between reactive astrocytes that were called A1 or unstimulated astrocytes. And then the most important part, what really everybody is interested is, are they killing the neurons? Because if that's true, we really should continue and better explore and dissect this mechanism and try to come up with something that blocks this killing. And so we use again, unstimulated astrocyte versus reactive astrocyte. In this case, is just a conditioned medium. And so we put the conditioned medium on iPS-derived neurons. We saw that there is an increase in the apoptosis of the cells, looking by caspase 3.7. So again, here you see conditioned medium from 3 independent astrocytic lines, and nothing happens when you have the cells with conditioned medium of unstimulated cells, or also when you have simply the 3 cytokines. So TNF-alpha, 1-alpha-C1q per se are not toxic to the neurons, but the conditioned medium from the reactive astrocyte is. And so, switching again to the other cell type, which is, very much important. And I feel, the two astrocytes and microglia are really connected because these cells are continuously doing this crosstalk. And, we talk about a lot microglia activating astrocyte, but I think it's bidirectional and both cells are really interacting in each other. So we also need to make microglia to have a complete story. The microglia protocol, there are several protocols that are available now. There were 4 protocols that came out in a few months. Started Rudolf Janisch in December, and then ours and Broughton Jones, and we have basically 4 protocols, and then a few more have came out during the years. And this is very important to tell you that this field is constantly evolving. It's not that nothing is written in the stone. We start with the protocol and then we keep modifying it as new things come out, new knowledge, so they get hopefully better and better. So in this case, we have to go to a mesodermal origin, because remember, the microglia are immune cells resident in the brain, but they migrate to the brain. They are originating in the yolk sac. So you first need to make these mesodermal cells, which is a nice monolayer from which after 2 weeks, you will see releasing. Microglia progenitors are just popping out and they are accumulating in the supernatant. So if you collect them, you can see that they are CX3, CR1, and CD14 double positive. And then you can use these progenitors either for a final maturation in vitro, or several groups have now used them and transplanting them into mice for having these xenotransplantations. And they have seen that the progenitors are able to really migrate and populate the mouse brain. And, the very important point, again, we said at the beginning, this is a model. Let's remember it is a model. This is definitely mostly sure for microglia cells, because every cell is the result of both genetic and environment. But some cells, the environment is really, really particularly important. And I would say that microglia, exactly for their role, which is the role of really responding to the microenvironment and change immediately, provide a very quick response to what happens. So these cells are changing very fast. And so a lot of people are skeptical about in vitro microglia because how these cells can be similar to the cells in the brain. And so certainly there are a lot of differences, and certainly the fact that we keep them in plastic alone changes them a lot. But again, what we need to understand, to try to discover is like what is that is similar to the primary microglia, and how we can use these cells, to still tell us something. And so, especially in the system that we want to use, where we have edited lines and we are changing one gene, we think it is still a very valuable tool because it's still helping us understanding the role of that gene in the cell, even if it's not that equivalent. And still, I think now that we have a lot of great imaging tools, super resolution, I mean, you can appreciate that certainly the cell have changed a lot from the iPSC, and they are ramified. They have these highly motile processes. They are moving around a lot, and so they really resemble microglia. They also are able to phagocytose. Again, we're using the same system, the PH-RODO conjugated synaptosomes. You can see them moving around and engulfing. We also gave them to eat some dying neurons, and this is a 3D rendering with Imaris, where you can see the cells is really trying to capture the apoptotic neuron. And so we have all these different cell types now, and what we have tried to do in the past years is making co-cultures to work them together. But instead of co-culture, another parallel way that a lot of people have been using is the organoid culture. So basically, you have the iPSCs in these specific media conditions that they will spontaneously form all the different cell types. All the different cell types, although always remember that have to be of the epidermal generation. So that does not include microglia, because as we said before, microglia comes from the mesoderm. So whenever you have those cortical organoids, you will have neurons, eventually astrocytes. For having oligodendrocytes, you need to provide them specific molecules. So NT3, PDGF, IGF, T3, they don't spontaneously make astrocytes, oligodendrocytes. But if you give them in a specific time frame, this is a protocol that Paul, again, our collaborator, published, and we helped him. So it takes a long time, and it takes over 200 days to be able to see MVP alignment and compact myelin. And of course, it's not in the entire organoids. I mean, we wish we had the fully myelinated organoids. It's not, it's still a low efficiency, but you do achieve you know, in a with very much patients, oligodendrocytes myelinating and wrapping the axons. So we have this protocol in house, and you know, you can predict, you know, what's next. We need to add the microglia to it. So we are. Other labs have already come out. A few papers came out. A few preprint have shown also different system where they're combining microglia to organoids. We haven't published our yet, but no, we talked to microglia progenitors. We tried at the beginning. We tried very simply to combine them in a dish, so they will just go down by gravity and they will attach and they penetrate. And you see here some sections of the organoid with the microglia, IBA1 and CD11C stain, and they penetrate and they persist in the organoids. We have single cell data at 6 months, so we know that they persist even after 6 months. And you can see them here. This was for a whole mounted organoid, and you can see, you know, the green is the IBA1 microglia cells in a 3D rendering below the TUJ1 neurons. And we are noticing that the morphology that they are inside the organoids looks more similar to what is called the resting microglia that you could see in a section of a brain. So really the environment seems to be very drastically important in shaping the cell. We have some very preliminary data on how the organoids change in the presence of microglia. So if you have the microglia and you let the organoid grow for 4 months, we are doing this MEA multi-electrode array studies, and we think it really looks like the microglia improves the oscillatory activity of the organoids, and so the organoids mature faster in the presence of microglia compared to organoids that are, you know, have less microglia. And obviously that, you know, will have a lot of important repercussions if, you know, want to study the role of microglia also in development and neurodevelopmental diseases rather than neurodegenerative diseases. And so, yeah, it's been almost 1 hour. Sorry, it went very long. But, you know, I try to give you a picture on an overview of the way, you know, we think as a stem cell biologist that IPS modeling can be important for CNS studies and can be used for improving our understanding of the human CNS cell biology. For example, you know, we saw the oligodendrocyte biology, the process of myelination, but also, you know, better understand astrocytes and microglia and the crosstalk between the cells and how we can modulate that in vitro. Long term, you know, something that it's not happening now, but we are moving toward that direction, is really the idea of the clinical trial in a dish. So being able to test compounds before we actually go to a clinical trial, test them in the patient cells, hopefully with the goal of better designing the clinical trial, so we can select before you know, the patients that would be most likely to succeed, and therefore increasing the results, because it's a huge problem. You know, most of 95% of drugs for CNS fail in this phase. And then also, you know, we said investigating pathogenic mechanisms linked to neurodegenerative diseases, and I'm particularly interested in the role of astrocytes and microglia. And so we have now this potential key drivers that we are studying in Alzheimer, and we hope that something interesting would come up from those studies as a new potential therapeutic approaches. And with that, this is my team that is all virtual, unfortunately. I hope we get soon a picture. Some of them started and I haven't even met them yet because they started through the pandemic. But it's really a lot of work, a lot of help from all the other people and myself. And thank you very much. And I'll take any question, even if we have short time. Thank you.
Ben?
Thank you so much. Your talk is really cool and the protocols are super interesting. So throughout your presentation, you kind of highlight both the opportunities and challenges of using iPSCs to create these various brain cells. So why do you think the iPSC-derived cells have such significant differences from in vivo cells? And do you think that you could use gene editing or other methods in the future to counteract some of these issues?
Yeah, no, that's an excellent question. So as I said, I think that the major point is, as we said, that the cell is a result of both environment and genetic. And so if you are changing the environment, you are inevitably changing also the cell. So a way that people are trying to make it better is, gene editing can help, but I don't think that's the way. We are more and more working in collaboration with bioengineers. And so try to recreate as much as possible the environment, the scaffold in which the cells are, and the other signals. And so the idea of using bioreactor, using matrices, for now we're using polyornithine laminin, but there's very cool works from bioengineering labs where they're really trying to recreate, scaffold, for example, from they take decellularized organs. For the brain, it's more difficult, but they've done it for pancreas, for cardiac cells. And so I think really what would really make the difference is to create the environment. And that's why organoids became so popular, because they are much better mimicking the environment. Here, you kind of let the cells spontaneously organizing, how's the eye they would. So it's we don't like calling them mini brain, because it's really not a mini brain. It's like really tiny piece that is not yet organized at all, like the brain. So this is another big point that I want to make. There are a lot of articles that came out in New York Times and a lot of different journals. How are these cells? Are thinking, their intelligence. I mean, we made a lot of progresses in reaching the diversity of the cells, but we're not there yet with the complex architectural organization of the cells. And again, I think it really needs an effort with the bioengineering primarily to make this happen. Really move very fast. They feel incredibly fast. If you consider how long the protocols are, because 10 years, it seems a long time, but it takes us 3 months to make a cell. And every time we have to wait till the end to be able to change something. That's the frustrating part. I don't know, Jim Wang, I don't remember what you did exactly when you were an SF, but it's all waiting, and then you reach at the end and like, oh, this didn't work, great. So you have to restart from the beginning. So 10 years, they just fly, honestly.
Thank you so much.
Thank you.
Jeff?
That's so true.
Hi. Thanks for a nice talk, Valentina. No, it's one of the nice things about waiting several hundred days for cultures. You could hang out in New York City, right? So, so I was curious about your microglia iPS cells. I mean, they do look a bit funny. I know that they may exhibit some features, but how do you differentiate these cells from other myeloid-derived cells in your protocol? Like, could they potentially be macrophages, or do they respond to LPS? Are you able to polarize them? Yeah, so the way that we show that they distincted by transcriptome profile, that may be good or not, but we bought or we isolated monocytes, macrophages, hepatic macrophages, and we compared by transcriptome profile, and we saw that they were different, and they cluster with primary fetal microglia. And this is the way that the field has, in general,
went. So every paper that comes up adds a more. So now we are enriching more and more the data. So hopefully, we are gonna be able to sort out better, which is the best protocol that really resembles the primary microglia. But yeah, they do respond to different stimuli. Definitely, they respond super well to LPS. And then I said, they secrete IL-6, RANTES, and TNF-alpha, IL-1 alpha, C1q. So all the things that were used for the reactive astrocytes. Yeah, the phagocytose.
So then in your protocol where you gave you induced reactive astrocytes with C1q and TNF-alpha and IL-1 beta. I think so. This reactivity is neurotoxic, but do we know what's causing it? I think you mentioned contact, but do we know why? I've seen this in the literature quite a lot, but I just don't know how it's killing neurons.
Yeah, so Shane has been working on, and it's, I think, very, very close to publish what is. So there is something that is secreted by the astrocytes that is directly killing the neurons.
Okay.
That there could be other mechanisms. So definitely what I'm interested in is now looking at all other features that change and now they may indirectly. And so for example, we are very interested in the metabolism. It seems that we have, David now is working on all this preliminary data. It changes a lot the metabolism of the cells. And so therefore, there can be a lot of indirect mechanisms for which the cells are less supportive. But what Shane is, really working on is to identify this molecule that is killing, that is secreted upon this this activation.
Thanks.
There was an end, Marina.
Yes, hi Valentina, beautiful work. I have two kind of related questions. So one, were you able to culture organoids from different patients with different conditions? You mentioned several diseases. Do you see the differences in cell populations in these organoids when you culture them? And second question is similar to that. When you do the astrocyte co-cultures, can you do, for example, astrocytes from a control patient, neurons from a diseased patient, and vice versa, and see the effect of the populations on each other?
Yeah, you definitely see that there are a lot of papers came out from other groups and not by us. My work on Alzheimer, it's still very early because we are still generating the lines, but my colleague Gist is working on Parkinson, and there they have familiar Parkinson lines with LRRK2 and different mutations, and so you absolutely see a phenotype. So you're able to see a phenotype in the neurons that are, so we've been doing all the mismatch astrocyte wild type with the LRRK2 mutated neurons and vice versa. And yeah, that's the beauty that you really find some phenotypes that hopefully we're going to be able to target.
And in organoids, do you see the difference between if you develop organoids from different patients, do you see the differences in cell populations and their development?
We haven't, no, we haven't, right, well, as of now, no, but again, we haven't done any organoid study with patient lines yet. We're only working with wild type. I mean, Paul has been working with leukodystrophies. So there you clearly see a huge phenotype on my oligodendrocyte lineage cell. So if you go with the leukodystrophies or these known mutations in the oligodendrocytes, you do see that. We haven't done yet organoids for Parkinson and Alzheimer, but this fact that the microglia seems to really change makes me thinking that that's what we hope to find, that all these genes that we are targeting in the microglia in our key drivers, we should find a difference in the neurons. But there are other labs. We are more interested in neurodegenerative diseases, but there are many labs, collaborators or none, that are working in neurodevelopmental diseases. And so for schizophrenia or autism, they do see some differences in the organoids. So, you know, there are a lot of unpublished and preprints. So all these studies are really coming up just because they take so long. You know, it's you know, some organoids. Paula Arlotta, you know, she's been Sergio, they published organoids 4 years old. But I know of also unpublished data on autism and schizophrenia where they do find differences. So they come up.
Thank you.
It's 33, so if people have to go, I just want to thank Valentina again for such a great talk. Thank you.
Thank you so much. And I look forward to the informal discussion with the students after the break.
Any last questions or?
Can I ask a very quick question, Valentina?
Sure.
Sorry, it's just, I think one of the things that, I mean, we could talk later anyway, but I just wanted to ask you quickly, you know, when you look at, when you culture oligos from patients, MS patients, primary progressive, secondary progressive, you know, what about patients, they have different treatment strategies? Do you see that this affects the iPS OPs oligos that you generate? You know, some are on disease-modifying drugs and others.
We can do a post-hoc analysis because I have all the data of what the patients have been treated with. So again, because now the number, the 30 lines that we have is so limited, I don't think we can draw any conclusion. But yeah, I would love to discuss with you more later because I don't, you know, yeah, it's hard.
Well, we could chat later.
Yes.
Well,
yeah.
Great talk. Thank you.
Thank you so much.
Thank you so much for coming.
So see, I use the same link. Well, I have the link in the calendar invite.
Yeah, and it's the same link for the meetings.
Perfect. Thank you again.
Thank you so much.
Thank you,
Bye.
Leslie. Bye.